Everyone, welcome back to Python/Linux. In this video, we're going to learn how to install bioinformatics software in a Linux environment. So most of you are upperclassmen if you're taking this for class, and you're just into bioinformatics if you're checking out on YouTube. So you've all probably heard of BLAST, right? So BLAST is on NCBI's website. It is a way to take an unknown sequence and check it against all known sequences in the NCBI database and tell you what gene it most, or DNA sequence, doesn't necessarily have to be gene, but what it most closely resembles, and it'll give you across multiple species and things like that. And so it's wonderful, except it's all in a web browser and it's hard to automate and things like that. So we're going to install it. We're going to install BLAST Plus in the Linux virtual environment in this class, or in this video. We're going to download some real sequences, and then we're going to index the genome and blast the sequences against the genome. So first things first, let me switch to, so you can see what I see. So there are different, for the most part we're using Ubuntu, so there are different like install managers that exist for different environments, and there's even multiple within Ubuntu. So there's apt and what's it, brew, homebrew I think is another one. There's a bunch of different like package managers. When we did, if you took my R class, there was the built-in CRAN libraries, right? Which was install packages, or there was BioC Manager, which was like two different ones. But there's quite a few for Linux as well. But we're going to use apt. So we're going to start by sudo apt install ncbi -blast. Actually, before I do this, let's go make sure we get into no, not downloads. Let's get into our directory. Classroom. So get to the my files part, and then do sudo apt install ncbi -blast plus. And as you can see, we get some information here. 5 newly installed. Do you want to continue? We're going to hit Y for yes. And it's going to start. Give us a progress bar and install everything. Oh, it's done already? Wow. Let's look away for a second. Okay, so let's do blast x-h. And so this is help for blast x. So we just did this to make sure everything's installed. You can see here's all the documentation. It's .h is the help file we talked about before. And so, okay, so now that we have blast, we're going to create a working directory in this my files directory. We're going to do mkdir. What are we going to call this? We'll do COVID, because we did the COVID, so we should have, see everybody's stuff. One called COVID. COVID Superlab, somebody else. I don't know what that is. Anyways, clear this, and leave it cleared. Okay, so I did make directory, and I just called one COVID. So we can also, so kind of like the apt command, there's multiple different download commands. There's curl, and there's wget, and there's some other ones too. And so let's cd into our COVID folder, and we should have nothing in there, which is good. So what we're gonna type, all right, I'm going to paste this and allow you to see what I've pasted here. Oh man, let's move this. Okay, that's the best way. I should just put this in like I'll type it out with you guys. All right. wget //ftp dot ensemble ensemble, or no e in it, genomes.org /pub /viruses /fasta /sars CoV-2 /DNA /SARS, the capital S, CoV-2 dot ASM9, oh my gosh, ASM9 85889 V version 3 dot DNA dot top level dot FA dot GZ. Oh, okay, it worked. Let me move this back over a little bit so I can. Okay, so oh, hold on a second. Okay, that's good. We're also, we need the protein sequence too. Peptide sequence. So get ready. We're gonna have another adventure here. Hopefully you guys can get this. I'll make it a little bit bigger so you can see what happened here. I guess not that big. All right, wget http. So it's gonna be, the whole beginning is gonna be the same. : //ftp dot ensemble. Remember, there's no e in ensemble. Genomes.org /pub /viruses /fasta /sars CoV-2 / instead, now this is where we're different from before. Before we did DNA, right? But now we're going to do PEP for peptides. And we're going to /SARS _ CoV-2 dot ASM 98588 version 3, like again, dot pep dot all dot fa dot gz. Beautiful. Okay. So both of those are saved now. I can kind of zoom back out a little bit so I can see a little more what's going on. Okay. So now what we want to do is we want to index the genome. And so what we're going to do for this is we're going to do this make blast db, which is a command from that blast package that we downloaded. And we're going to say in SARS-CoV _2.asm 985889v3 .dna .top level .fa, and then we're going to do db type nuke, or say out SARS-CoV-2 genome. I misspelled my file because it's saying it doesn't exist. Let me make sure oh, you know why? Because I forgot to gunzip my two files. So we're gonna gunzip SARS. Oops. I'm gonna gunzip the SARS pep file. I'm gonna gunzip the SARS DNA file. So now if we look, we have just the FA files, so not GZ anymore. Now I'm gonna go back by hitting up to my make blast db command and run that. Beautiful, and that is done. And then I'm just gonna hit up again, and I'm gonna change this from DNA to PEP, because that's the only difference between the two files. And I'm gonna run this on the PEP file. Oh wait, maybe it has a different name. SARS-CoV-2 PEP top level FA. Oh, it's because I did database type. It should be not nucleotide. It should be PEP, right? Wait, why is it giving me this now? Error. Oh, it should be. I have PEP. Wait, illegal value. Expected nucleotide protein PEP. DB type. Okay, let's try. Maybe it's supposed to be protein. You know, we said it's a peptide sequence. OT. What is wrong? SARS. Alright, let's do this. Let's list our files, and then let's make sure these match. Make blast database in SARS-CoV-2 pep.all.fa, not top level. We have to change one more thing. All.fa. And now let's change it back. Let's try pep again before we do this protein thing. Okay, so it still doesn't like me using pep for some reason. Alright, let's try protein. We did it. Success. Okay. What are we at? 14 minutes. Alright, let's keep going. So let's blast our protein sequence against our nucleotide sequence. So for this, we'll need tblastn, which stands for nucleotide. So we're going to do tblastn db SARS-CoV-2 genome query. We're going to say our SARS we're gonna tab, so I don't have to type in all that stuff, and then we're gonna select our PEP, and then tab again. E value, so this is our significance value for finding matches. So we're gonna say 1 to the negative 50, and then greater than symbol, we're gonna say last underscore first SARS-CoV-2 dash hit table dot t last n. So that's our output, or what we're piping into, or amending, appending. That's probably a better. So this puts it into a file, right, instead of displaying everything. Too many potential arguments. Offending value. Okay, what did I do? E value query. Oh, I think best database. This probably was not supposed to have a space in between here. No last index file path. What did I call it before? SARS-CoV-2 genome. SARS-CoV-2 genome. What's the LS here? SARS-CoV-2 genome. The protein interactions. Okay, error, too many position arguments, blah, 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 blah. No alias or index file find for nucleotide database in search path. Make blast database. Can we get the nucleotide one to work? SARS-CoV-2 genome. In oh, why didn't it run before? Okay. Okay, now it worked. For some reason, the first time I ran, I'll show you what I did. So we ran this make blast to make this protein thing that we stumbled around on, make blast database. And we said take this peptide file and then make these protein index files. Well, we also earlier did that for nucleotides, and I thought it ran, but if I reran this code, make blast database in our SARS DNA top level FA, FASTA file, our database type nucleotide, and our SARS-CoV-2 genome, then it actually made more files, these nucleotide files. So here we have protein or peptide files, and here we have nucleotide files. But I thought we already ran that command, so I don't know why that is. But it works now. So then after that, I ran this tblast command that we were doing before, and that worked. All right, so let's say C. Okay, let's run this again. But let's add, in front of query here, let's add this out fmt6 argument. And so actually, let's not do that yet. Let's do head of last this table that we did. Okay, so it just shows the references. Let's do tail of the last thing we saw. So okay, let's do less of it. Okay, so we can kind of jump through it here, you know, keep hitting our space bar. It's getting late. My words are escaping me. And we can keep going through this. But as you can see, it's matching up our query, which is what we entered, which is this version 3 that we had, against the subject, or what's in the database. And it gives us scores. We see it says we have 99% identity, 0 gaps for this alignment, protein alignment. This is a different assembly of COVID. And so you see that we have good matches for everything. And remember, if you want to get out of this, you just hit, oops, I want to go to, Q. This is Q. Oh, okay, yeah. I can remember. Q, or Q, or Q, or Q, or ZZ. Does accent. Okay, so that is our Q. Jeez. Okay, so we're out. Okay, now I said we want to do this out format 6 is a way of changing the format. Out. There we go. Out format space 6. So now if we do less of actually, what is that called? Oh, it's right there. Last. You can see that we have a different format here, right? So we have our alignments and then all of our scores lined up that way. Instead of having every piece of alignment, this is just kind of the summary of our alignments. Okay, and then we're gonna stop right there for now. We're at 23 minutes, perfect. Okay, so we stumbled through that a little bit. You know, I made typos too. But hopefully you got a chance to get all this to work, and see how you would blast something. So we know we downloaded the COVID sequence, right? And then we're like, blast it against other COVID sequences to see percent identity. But for instance, the first people to isolate COVID didn't know it was COVID, right? So they then sequence it and then they blast it to see what it's supposed to like. So there are other COVID or SARS type viruses, right? And so they find that SARS-CoV is similar to other SARS viruses, and they can kind of get an idea of what type of virus it is and what it does, whether it's a DNA virus, an RNA virus, etc. So that would be the utility in doing a blast like that. But we're just going through the motions of learning how to download. The whole point of this was to show you that you can download these bioinformatic tools into the terminal and run them. And so we played around with the COVID genome for that. So without getting too much more into it, I think I'm going to make another short video and we're going to blast some human stuff real quick. I just don't want this to go too much longer. And then we'll be on to our next topic. So thank you for watching. Hit like, hit subscribe if you're watching on YouTube. I appreciate you